In Chapter 10, Segment 3, most cells in the primary visual cortex are tuned to particular spatial frequencies. It says that the spatial frequency filter model is a model of pattern analysis that emphasizes Fourier's analysis of the visual stimuli. Spatial frequency of a visual stimulus is the number of light-dark or color cycles that the stimulus shows per degree of visual space. A complex visual pattern or scene can also be analyzed by the Fourier technique. To reproduce or perceive a complex pattern or scene accurately, the system has to handle all the spatial frequencies present. When high frequencies are filtered out, the small details and sharp contrasts are lost. When low frequencies are filtered out, the large uniform areas and gradual transitions are lost. Sensitivity to the subsequent gratings were reduced briefly at the particular frequency to which a person had adapted. An example is the Mona Lisa. Sometimes the model seems to be smiling, and other times she isn't. This could be due to the differences in spatial frequency. The low spatial frequency components of the mixture make it look like if she is smiling, but the high spatial frequency components give her a sad expression. An area V1 is involved in the formation of mental images. It says that imagined objects activate regions that correspond to the retinal topic mapping of V1. In neurons in the visual cortex beyond area V1 have complex receptive fields and contribute to the identification of forms. It says that area V1 is only a small part of the portion of cortex that is devoted to vision. Area V1 sends axons to other visual cortical areas, including areas that appear to be involved in the perception of form. V2, V4, and the other inferior temporal areas. Anatomical, physiological, and behavioral investigations with macaque monkeys reveal at least 32 distinct cortical areas that are directly involved in visual function. The visual areas of the human brain have been less thoroughly mapped than those of the monkey brain. The areas that are largely or entirely visual in the function occupy about 55% of the surface of the macaque cortex and about 30% of the human cortex. Area V2 is adjacent to V1, and many V2 cells can respond to illusory contours. Area V4 receives axons from V2, and the cells that give their strongest response to the sinusoidal frequency gratings and give even stronger responses to concentric and radial stimuli. They also respond strongly to wavelength differences. Area V5, also known as the medial temporal area, appears to be specialized for the perception of motion. Most of the cells in inferior temporal visual cortex do not require natural objects such as a face to activate them. They require moderately complex shapes, sometimes combined with color or texture. In area V1 is organized in columns. It says that the primary visual cortex has separate representations for at least 4 dimensions of the visual stimulus. Location in the visual fields with larger mapping of the central region of the visual field than of the periphery is the first one. Then it's ocular dominance, orientation, and color. Ocular dominance columns is a region of the cortex in which one eye or the other provides a greater degree of the synaptic input. They are arranged in ocular dominance slabs. A slab of visual cortex is about 0.5 millimeters wide, in which the neurons of all layers respond preferentially to the stimulation of one eye. Ocular dominance is clear in layer 4, where each cell is monocular, responding to only one eye. Up to the age of 3 or 4, infants are captivated by stereograms. The cortex up to that age, information from the 2 eyes. Orientation columns is a column of visual cortex that responds to rod-shaped stimuli of a particular orientation. All the cells may be tuned to upright stimuli, and adjacent columns all respond to another orientation, perhaps at 10 degrees from the vertical. There are columns of cells in V1 that respond to particular spatial frequencies. In color, vision depends on special channels from the retinal cones through cortical area V4. It says that there are 4 stages of the color perception. The cones, the retinal receptor cells that are specialized to respond to certain wavelengths light, receive visual information. Then the information is processed by neurons in the local circuits of the retina, leading to retinal ganglion cells that are excited by light of some wavelengths and inhibited by light of some other wavelengths. Next, the ganglion cells send the wavelength information via their axons to LGN, mainly in the parvocellular layers. And then from there, this information goes to area V1, which is related to other visual cortical areas where additional stages of color perception take place. In colors created by the visual system, it defines hue as a dimension of light perception, varying around the color circle through blue, green, yellow, orange, and red. These hues appear different due to the reflected light that reaches our eyes and can vary in wavelength. In 0.5% of females and 8% of males, some of the color distinctions are less striking or absent. Color blindness can be caused by brain lesions or the congenital absence of specialized receptors, but in humans it is extremely rare. Animals have a different degree of color vision. A patch of illuminated light by a particular wavelength has various different hues, depending on factors such as the intensity of illumination, the surrounding field, and prior exposure to a different stimulus, which is why perception of hue does not depend on a certain stimulus. Hue perceived at a particular point is strongly affected by the wavelength and intensities. In color perception requires receptor cells that differ in their sensitivities to different wavelengths. It defines the trichromatic hypothesis as a hypothesis of color perception, stating that there are 3 different types of cones, each excited by a different region of the spectrum and each having a separate pathway to the brain. This was proposed by physiologist, physicist, and psychologist Hermann van Helmholtz. He predicted blue-sensitive, green-sensitive, and red-sensitive receptors would be found, and each would be tuned to part of the spectrum and have a separate path to the brain. Ewald Turing proposed the opponent process hypothesis. This hypothesis is a theory that the color vision depends on systems that produce opposite responses to light of different wavelengths. Each cone of the retina has 1 out of 3 types of pigments and don't have the narrow spectral distributions Helmholtz thought. The human visual system does not have receptors that are sensitive to only a narrow part of the visible spectrum. 2 out of 3 retinal cone pigments show some response to light at basically any wavelength. The spectral sensitivities of each 3 cones differ from each. The nervous system detects and processes these different between cones to extract the color information. Peak sensitivity at 420 nanometers is known as short, while peak sensitivity at 530 nanometers is known as medium, and peak sensitivity at 560 nanometers is known as long. Actuality is much lower with short wavelength illumination than in other parts of the visible spectrum. Photopigments are inheriting genes that cannot produce functional opsins.